Hey everyone, we're going to go ahead and go through the muscles and muscle tissue chapter. It's a pretty long set of slides, so I apologize in advance. There's also some YouTube videos I'm going to try to kind of incorporate into this, so we'll see how that goes. Bear with me. I'll try to make it as smooth as I can. So let's go ahead and cover some terminology that you may hear me say, and just in case you don't know what it means, I want to talk about it now. So um, you're going to hear me use the word muscle fiber. And when I'm talking about skeletal muscle and smooth muscle in particular, although for the purposes of this chapter, we'll just be talking about skeletal muscle. But when I use the word muscle fiber to describe skeletal muscle or smooth muscle, you can replace the word fiber with the word cell. So muscle fiber means muscle cell. And it's just called that because of how long and skinny these muscle cells are. If you think about some of the muscle cells in your sartorius muscle, I mean, you could have a cell that's a couple feet long. If you think about the origin and insertion of sartorius and the fact that one muscle cell will go from origin to insertion minus the length of the tendons, then that's one cell and it looks like a fiber. So we call it a muscle fiber. You'll also hear the words myo, and I think you could probably guess that that means pertaining to muscle. So myoglobin is a molecule that's like hemoglobin, but within muscle cells. So we call it myoglobin. You'll also see the word sarco. Sarco means flesh, which for all intents and purposes means muscle. So if you see the word sarcoplasm, that's the cytoplasm of a muscle cell. If you see the word sarcolemma, that's the cell membrane of a muscle cell. So muscle cells are so unique that they just have all this term, their unique terminology. Neurons are the same way. We have a neurolemma and stuff like that. So anyway, so myosarco means muscle, muscle fiber means muscle cell. So we're going to look at the three different types of muscle tissue. So we have skeletal, cardiac, and smooth. So skeletal muscle is the longest. These are very long muscle cells, and that's just due to the fact that they're cylinders and that skeletal muscles are much longer than, smooth muscle would be or your cardiac muscle would be. So we call them fibers for that reason. They are striated. Now, what that means is, I wonder why, oh, here we go. So it's not referring to the fact that they're long fibers. Uh, that's not what striated means. What striated means is that if we look at them in cross section, you can see that there's these little stripes, these little striations. And that actually tells us a lot about their structure and function. It tells us that they're using myosin and actin cross bridging to create muscle contraction. And you'll learn why that is later. Skeletal muscle is voluntary. You decide what your skeletal muscles do. It's not like your heart or smooth muscle, which are involuntary. And it's very powerful. With great power comes great. No, I'm kidding. Because it's powerful, it's also easily fatigued. So it's a trade-off. Cardiac muscle isn't nearly as powerful, but it also isn't as easily fatigued, which is nice. You don't want your heart to get tired. It's also highly adaptable, and all that means is it can produce many different levels of force, many different grades of force. Cardiac muscle is also striated, so it uses a similar mechanism of contraction as skeletal muscle. However, it's involuntary. And this next bullet point here, connected by intercalated discs, that's a fancy way of saying that there are gap junctions between these cells, and that really changes the way that heart muscle works compared to skeletal muscle. Basically, it's like a light switch. It's either on or off. And there are some different grades of contraction, but it's not like skeletal muscle where we can really fine tune. We can lift a paper clip, we can lift a bowling ball. We're able to just really adjust our force, and we're able to determine which fibers are turned on because each fiber is insulated from the fiber next to it. It's not the case with heart tissue. They're all electrically interconnected. So if one cell contracts, they all contract. That contraction, that electrical activation force spreads. Then we have smooth muscle. Smooth muscle is found in walls of hollow organs, so stomach, intestines, bladder, blood vessels. These are elongated cells, so we still call them fibers. However, though, if you look, there's no striations. And that's where their name comes from, smooth muscle. So it's involuntary. You can't think really hard about digesting your cheeseburger faster. It just does what it does. And it produces very slow, weak, but sustained contractions. It doesn't fatigue easily. You can't wear your stomach out. Okay, so let's talk about some special characteristics of muscle tissue. Muscle tissue is excitable. What that means is it can respond to an electrical force. It can actually conduct an electrical current, which we call an action potential when we're talking about nervous system electrical impulses and muscle electrical impulses. We call it an action potential. So it is excitable. That's thing one. Thing two is that it's contractile. And that doesn't mean that it can forcibly shorten necessarily. It just means that it can produce force. It can produce force. So it can produce force while lengthening. If you've ever set something heavy down, let's say you're doing a bicep curl and you lift the weight up, and the weight's up high. You flexed your elbow, and then you slowly lower it back down. Your muscle's contracting, but it's lengthening. So contractility just means producing tension. It doesn't mean shortening. Although you can also produce tension and shorten a muscle as well, obviously. So you just got to separate those two things. Muscle tissue is extensible. That means it can be lengthened, and that's easy to see. As soon as you flex your elbow, your triceps have to lengthen, and then when you extend your elbow, your biceps have to lengthen. So they're extensible. And then we have elasticity, which does not mean stretchy or can stretch. That's really what extensibility means, right? If you take a rubber band and you stretch it way out, that's extensibility. When you let go, it snaps back to its original shape. That's elasticity. So muscles are both, just like a rubber band. What does muscle do? Obviously, it does movement production, maintains posture, body position, joint stabilization. It also produces a lot of heat. That's why we shiver when we're cold, because that helps produce heat. It also protects our organs. Obviously certain muscles more than others. Our abdominal muscles protect our abdominal contents. Valve formation, like our ileocecal valve, pupil constriction. There's sphincter, the pyloric sphincter, for example. All different types of functions, but primarily it's movement production, whether that's movement of blood, movement of food, or movement of bones. Movement production is its main function. So what does striated mean? Well, what it means is that there are alternating bands of light and dark regions in a muscle cell, within the cell itself, not between cells. Okay, so there's alternating bands of light and dark, and we're going to learn why that is in a second. Andrew is pondering an exam question. Oh, sorry, I can't yawn during these. You're gonna start yawning. Andrew is pondering an exam question that asks, which muscle type has elongated cells and is found in the walls of the urinary bladder? How should he respond? Smooth muscle. Smooth muscle is found in the walls of hollow organs. All right, so now we're going to start looking at gross anatomy, and then we'll kind of go down to microscopic anatomy. So each muscle is a discrete organ. It has its own nerve and blood supply, and it's composed of multiple different tissues, so it is a discrete organ. We name these tissues, these connective tissue sheaths, based on their location. So if we were to touch a muscle in lab, we're actually not touching muscle fiber. We're touching a connective tissue wrapping. Picture shrink wrap or gift wrap on top of a package. That most external, most superficial connective tissue sheath is called the epimysium. And you can see that in the diagram here. Then we have the perimysium and fascicles. So if you've ever had a nice steak, or maybe like some shredded beef or shredded chicken, maybe you've shredded chicken. That's probably the best example. So if you ever shredded chicken by hand, you kind of pull it apart, right? And it pulls apart in these little stringy bundles. And those bundles are called fascicles. Those are groups of muscle fibers held together by connective tissue. So we call that we call that connective tissue sheath that holds the fibers together. We call it the perimysium. and then we call the bundles fascicles. So right here is a fascicle or a fasciculus, right here, and then you can see it's a collection of muscle fibers inside. Which brings us to the smallest, deepest connective tissue sheath. Here is one muscle fiber. Don't get confused. That looks like more muscle cells inside. They aren't. Those are called myofibrils. This right here is a muscle cell. You can see the nuclei of that muscle cell sticking out. The connective tissue wrapping around this cell is called the endomysium. So epi, peri, and endomysium. The only one people tend to get confused is epi and peri. Peri means surrounding, so they feel like it's the outside. Epi means upon, so epi is actually the outermost wrapping. Perimysium bundles fibers together in fascicles, and endomysium surrounds the individual muscle cell. Don't worry about attachments. All right, so here we are on microscopic anatomy. So what we're going to start with is just kind of some general regions of the muscle cell. So this is a little piece of a muscle cell. Of course, it'd be much longer than this, but you can see that inside there are tons of these little contractile rods called myofibril, and we can see some nuclei. So muscle cells are multinucleate just due to how large they are and the demand for protein production. So muscle cells are multinucleate cells. You can see the sarcolemma peeled back here. That's the cell membrane. And then within the cell membrane and outside of the nuclei, we have what's called the sarcoplasm. And this is just cytoplasm. In addition to the typical organelles, you're also going to find structures called glycosomes. These are vesicles containing glycogen, which is a fuel source for the mitochondria to produce ATP. And then we have myoglobin. Now, myoglobin is just essentially hemoglobin, so it binds oxygen, and it's contained within muscle cells. So you have a fuel and an oxygen source within the muscles, both of which are necessary for aerobic respiration. Here's where it starts to get we start to have to use our imagination a little bit to visualize what these structures look like. So now we're going from looking at the muscle fiber to looking specifically at this myofibril, okay? So where's the best place to start? Okay, let's start with this idea. So a myofibril is a series of sarcomeres in series. It's sarcomeres in series, like a chain. So this region right here, including the Z disc, sorry. So between here and here is called a sarcomere. That's the smallest contractile unit of a muscle cell, of a myofibril specifically. So this is a contractile unit. It is able to forcibly shorten. So these structures here, the thick and thin filaments, are able to pull the Z discs together. And they might shorten, less than a millimeter, right? But then if you put thousands of them in series, and they all shorten a millimeter, that's thousands of millimeters of shortening, right? So you can produce a lot of movement through this mechanism. So let's see, striations, sarcomeres, and myofilaments. Okay, so let's zoom in a little bit. So this is just kind of a diagram of what a few sarcomeres look like. It's really one full sarcomere and two partial sarcomeres. Now we're going to zoom into one sarcomere here, and this is what we see. Now we're looking at this level. So what we see are 3 red, what we call thick filaments, and 1, 2, 3, 4, 5, 6, 7, 8 thin filaments in this bluish color. You can see that there are regions where they both overlap, right? So the thick and thin filaments are overlapping right here. That's going to look darker on the muscle cell. So you see how there's these dark bands? It's because in that region, there's overlap. And then we have this region here around the Z disc where it's just thin filaments, That's going to appear lighter. Then if you look here, you can kind of imagine, it looks a little bit darker right there. And then, this region that's just the Z disc, just thin filaments only, that looks a little bit lighter. So we call the darker area the A band, which is nice because there's an A in dark. And we call the lighter area the I band, and there's a I in light. So that's pretty easy to keep track. So now you can see dark, light, dark, light. That's where the striation comes from. It comes from the way that these filaments overlap one another. Okay, don't worry about H zone and M line. I just want you to understand why we have these alternating dark and light bands. And I want you to understand that the sarcomere is the region between two successive Z disks. Okay? All right, so now let's look at the filaments themselves. So first of all, muscle fiber, that's the muscle cell. Myofibril, those are the contractile rods within the muscle cell. And then myofilaments are the actual working structures that cause contraction within the myofibril. So they're even smaller than the myofibril. So there's two of them. We have thick filaments and thin filaments. I often accidentally call these myosin filaments and actin filaments, but I think that's just a good a name because that's what they're made out of. So the thick filaments are made of myosin. There's some other things like titin and stuff, but myosin is the primary structural component of the thick filament. And here you can see a single myosin molecule. All right, so we have this long tail that's interwoven because there's actually kind of two heads with their tails interwoven. And then there's a flexible hinge region and two myosin heads. Those myosin heads are able to walk. So if you can picture them flexing and extending, and then you put thousands of them together, you have this structure that under the right circumstances can pull on something. If you picture these heads being able to flex and extend, they can pull on something. And what they're going to pull on is this thin filament here, which is made up of troponin, right here, troponin complex, tropomyosin, and actin. So we're gonna talk in some depth about all three of these. For now, I just want you to know that they exist. The actin subunits create the actual rod, the structural rod. Troponin blocks the myosin binding site at rest. I'm sorry, tropomyosin blocks the myosin binding site at rest. And then troponin is able to move tropomyosin out of the way. None of that makes sense right now. If you haven't learned this before, I'm just kind of trying to foreshadow a little bit. We're going to watch a video about how this all works. But when a muscle contracts, what happens is the myosin, the thick myosin filaments, pull on the actin filaments, and they pull them closer to this middle line called the H zone. And when that happens, the Z disks get pulled towards one another, and the sarcomere shortens. And if you get thousands of sarcomeres shortening at the same time, you get a muscle contraction. Okay? And here, this video is just kind of revisiting the same thing. So we have myosin. Myosin is able to pull the thin filaments towards the middle here, toward the H zone. And then, again, this is just for reference what these filaments are made of. We're going to learn about each one of these, okay? Actin's really just the structural component of the thin filament. Tropomyosin and troponin, these are going to become very important in terms of how the muscle actually contracts, okay? But just hold on to that. We're going to watch a couple really good videos later. Okay, so before we get into the video, we kind of need to understand a few more structures. We need to understand the role and the existence of the sarcoplasmic reticulum and what t-tubules are. So this is, actually, I really like this diagram. So this here is the cell membrane, okay? And then, so we're kind of looking inside the cell. We're looking at, looks like 5 myofibrils right here. And there's two structures of interest. One are these tubes, these yellow tubes right here. All right, sorry. So we have these yellow tubes right here, and these tubes have extracellular fluid within them because there's a pore on the surface right here. You can see that. We're inside the cell, but you can imagine extracellular fluid could flow down into there, right? So all of these tubes contain extracellular fluid, even though they're inside the cell. And they surround every Z disc. Every Z disc has a T-tubule around it. And then on either side of the T-tubule, we have this big blue structure here, okay? And then we have all these tubes coming off of that blue structure. This is bad drawing. You know me. All right, so the idea here is that this blue structure is called sarcoplasmic reticulum, and it stores calcium. And then near every T-tubule, the sarcoplasmic reticulum forms this bigger pouch that's adjacent to the T-tubule. Those are called terminal cisternae. So you have two terminal cisternae and a T-tubule on top of every Z disc. And then we call this structure, these three things together, we call a triad. So it's a little bit easier than saying a t-tubule and two adjacent terminal cisternae. So we just say triad. Okay, but the big thing here to know is that the t-tubule is continuous with the extracellular fluid. It contains extracellular fluid, and it's an extension of the sarcolemma. So if we can create some type of electrical current on this cell membrane, it will naturally be conducted down the t-tubule. So you can almost think of the t-tubules like copper wires that conduct electricity. And then this blue structure here, the sarcoplasmic reticulum, stores calcium. And it releases calcium when exposed to a certain voltage. So you may kind of start to be imagining what might happen. If we get an electrical stimulus here, it's going to travel throughout the muscle cell because of this copper wiring, which isn't copper wiring, of course. It's a tube filled with extracellular fluid. But anyway, it conducts that electrical signal into the muscle cell, and it opens gates in the terminal cisternae which allow calcium to release. And we're going to learn soon that calcium release is what causes muscle contraction. Okay, so one last slide here, and we're going to go watch 3 videos in a row. And you know, I've been meaning to change this for a while. Let me give you guys a little help here. So there's a few videos. So let me jump a few slides forward. You see there's this activation and excitation contraction coupling slide. There's a neuromuscular junction slide. There's a action potential across the sarcolemma slide and excitation. There's all these slides and steps here. I just took it out of your book. I wrote it the way the book explains it for your reference. However, we are just going to watch 3 videos in a row that I think explain it much better. And we're going to talk about the events at the neuromuscular. If you want to write this down on a piece of paper, we're going to break it down in 3 ways. The events at the neuromuscular junction, and then we're going to look at excitation contraction coupling, and then we're going to look at cross-bridge cycling. We're going to break it down into those 3 steps. I just think it's simpler. I hate to have it 2 different ways, but one's out of your book, so when you go back and study, you kind of can see, you know, what I wanted you to read. But really, I'm going to be talking about the key events in these 3 videos, and if you can understand those key events, there's not going to be any questions on the test you can't answer. So apologize if that's a little confusing. Let's just watch the videos and see how you feel afterwards. So anyway, last slide before we do that, I just want to address this model here. This is the model that explains how muscle fibers contract, how skeletal muscle fibers contract. And we've already addressed this to an extent, but basically in a relaxed muscle fiber, there's only a little bit of overlap. Okay, you can see right here, there's only a little bit of overlap between the thick and thin filaments. But when we activate this muscle, when we cause contraction, what happens is that the thick filaments tug on the thin filaments and they bring them in. And that results in a shorter sarcomere. I'm trying to draw one of those little brackets. Oops. So you can see this is a much shorter sarcomere than this one. And that happens because the thick filaments pull the thin filaments towards midline. Okay, and we're going to watch the video to see how that works. Okay, so this first video is going to be on the events at the neuromuscular junction. So basically, and first of all, don't judge me. I have all these like remote control airplane videos over here. my daughter likes to watch these. We watch rocket launches and we watch Mickey Mouse Clubhouse and Peppa Pig. So it's a variety over here. Anyway, so basically there's going to be 3 videos, and so what we're looking at is we're looking at how the electrical signal gets from the nervous system to the musculoskeletal, or to the muscular system. So how the stimulus gets from the neuron to the muscle fiber. That's the first step. And then we're going to watch a video on how do we take that electrical signal and turn it into a physical response. And that process is called excitation contraction coupling. So that will be video 2, excitation contraction coupling. And then video 3 is, okay, we've triggered the contraction. How does the contraction actually occur? And that's going to be called cross-bridge cycling. So you can reference the slides that we have in our PowerPoint. There's a slide for each of those. But But really, I'm just going to talk through these 3 videos, and I'll try to think of maybe some potential quiz questions along the way to prep you guys for. And we'll get through this together. Just a heads up, it's a lot of information, so hang with me. So this is part 1, which is the events at the neuromuscular junction. I'll kind of chime in here as we go.
Nerve impulses, also known as action potentials, travel from the brain or spinal cord to trigger the contraction of skeletal muscles. An action potential propagates down a motor neuron to a skeletal muscle fiber. The site where a motor neuron excites a skeletal muscle fiber is called the neuromuscular junction. This junction is a chemical synapse consisting of the points of contact between the axon terminals of a motor neuron and the motor end plate of a skeletal muscle fiber. The events at the neuromuscular junction.
Okay, so I just want to stop here and try to explain what we're looking at. It may be obvious to you, but it might not be, so I want to talk about it. This kind of bulbous looking blob here, this is the end of the neuron. It's called an axon terminal. There's a lot of them. There's not just one. There could be thousands potentially, but we're just going to look at the junction between one axon terminal and a muscle fiber. So we call this junction the neuromuscular junction. This yellow blob here is the axon terminal, so it represents the neuro in the neuromuscular junction. And then this yellow area here is the sarcolemma. So that's the cell membrane of the muscle cell. So it's the neuromuscular junction. Now what they're also showing you, which is really cool, is the t-tubules. So you can see that the t-tubules are continuous with the sarcolemma. Extracellular fluid can just flow right in. And then you can see it there wrapping around the z-discs. So this area here is a sarcomere, right, because it's between the 2 z-discs. And then you can see all the sarcoplasmic reticulum and terminal cisternae. All we're really going to be looking at in this video is the events that occur right here in this cleft between the axon terminal and the sarcolemma, which is called a synaptic cleft. Typically that refers to, a neuron-neuron synapse, but it applies here as well. So we're going to be looking at the chemical released from the axon terminal and its effect on some receptors in the sarcolemma. So let's check it out. Now, she's going to list, I think, 7 steps. I'm not going to say, like, hey, what's step 3 of the events of the neuromuscular junction on a quiz, so don't stress about that. I'll kind of do a recap of what I consider the key events, and I just want you to have a conceptual understanding of them.
Occur in 7 coordinated steps. Step 1. An action potential travels the length of the axon of a motor neuron to an axon terminal. Step 2. Voltage-gated calcium channels open and calcium ions diffuse into the terminal. Step 3. Calcium entry causes synaptic vesicles to release acetylcholine via exocytosis. Step 4. Acetylcholine diffuses across the synaptic cleft and binds to acetylcholine receptors, which contain ligand-gated cation channels.
Oh, I love physiology terminology. Ligand-gated cation channels. So far, really all that's happened, if we want to be honest, instead of trying to be overly complicated like all physiology videos, is that an electrical signal traveled down the neuron. That electrical signal triggered acetylcholine release, and now acetylcholine has floated from the axon terminal, or diffused rather, over to receptors in the sarcolemma. Now, ligand-gated cation channel, all that means is it's a chemically gated positive ion channel. So what that means is if a certain chemical, in this case acetylcholine, binds to this channel, it will open and allow a positive ion to travel through it. So what we're going to see is that sodium is going to enter and potassium is going to exit. Those are both positive ions. So imagine something for a second. I wish I could draw on this. Imagine, well, we don't have to imagine, we know that sodium is very highly concentrated outside of our cells in the extracellular fluid. So up here is extracellular fluid. Down here, you can only see a little piece is intracellular fluid. So in this extracellular fluid, there's a ton, scientific term, a ton of sodium, a very high concentration of sodium. It's a 10x differential between outside and inside. So if this channel opens and allows sodium to travel through it, where's the sodium going to go? It's going to go in. The sodium is going to travel down its concentration gradient into the cell. Sodium is a positive ion. So if we get a huge wave of positive ions moving into the cell, we've created a current. That's an electrical current. And now that we have electrical current entering the cell, this is getting a little beyond this chapter. It's kind of getting into the nervous system chapter, but once we get a flow of positive ions rushing into the cell, that's going to trigger more cation channels to open. There are voltage-gated cation channels and chemically gated cation channels, and it's a domino effect. So we get this wave of electrical current, and they're going to represent this wave of electrical current as this magical yellow glow, but we know it's really the opening of cation channels. So I'm just going to back up a little bit because I've been rambling, and let's watch this again from the top and see what happens.
Acetylcholine diffuses across the synaptic cleft and binds to acetylcholine receptors, which contain ligand-gated cation channels. Step 5. These ligand-gated cation channels open. Step 6. Sodium ions, shown here in red, enter the muscle fiber, and potassium ions, shown here in blue, exit the muscle fiber. The greater inward flux of sodium ions relative to the outward flux of potassium ions causes the membrane potential to become less negative. Step 7. Once the membrane potential reaches a threshold value, an action potential propagates along the sarcolemma.
Look at that beautiful wave of electricity. So this is such a loaded statement. Once the membrane potential reaches a threshold value, an action potential propagates along the sarcolemma. Ooh, what in the world does that mean? Well, what it means is in this area here, we have chemically gated sodium channels, ligand gated cation channels, you could call them. So they open in response to acetylcholine, and sodium rushes in. At some point, though, so much sodium has rushed in that the electrical value here, the membrane potential, it's called, the difference between the inside and the outside, becomes so low, around negative, well, I think it's around negative 55 in muscle, for sure in neurons it's negative 55. But once that differential becomes negative 55, it triggers voltage-gated sodium channels to open. So let's watch it again.
The threshold value.
Okay, so that glow is representing a threshold value. And then what we're going to see is then, once that threshold value is reached, voltage-gated channels open, and it's a domino effect that causes this wave of electricity to spread.
An action potential propagates along the sarcolemma. Neural transmission to a muscle fiber ceases when acetylcholine is removed from the synaptic cleft.
Okay, we can leave it there. So that's it. I mean, let's look at it again. So acetylcholine's released. Okay.
Mitosis.
Released. It diffuses across the synaptic cleft.
Step 4. Acetylcholine diffuses across the synaptic cleft and binds to acetylcholine receptors, which contain ligand-gated cation channels. Step 5. These ligand-gated cation channels open.
Okay, so this is so slow. Let me just recap. The action potential reaches the end of the neuron. The neuron releases, or rather the axon terminal releases acetylcholine. Acetylcholine diffuses and binds to acetylcholine receptors. Those acetylcholine receptors open a sodium channel and sodium rushes in. Okay, so that wave of sodium rushing in triggers an action potential, which is an electrical signal that spreads throughout the muscle fiber. So that makes sense. think of questions like which chemical is responsible for diffusing across the synaptic cleft and opening cation channels? Acetylcholine. What happens when cation channels open in the sarcolemma? Sodium rushes in and triggers an action potential. So the questions will be kind of basic like that. And we'll have a practice quiz next week and stuff to get ready for the final. So don't stress too much. Let's go on to the next video, and I think I can switch tabs without my recording stopping. Let's see. Okay, hopefully you're still with me. So now we're looking at part 2, which is excitation and contraction coupling. We saw excitation, we saw that yellow glow, that action potential spreading across the sarcolemma and the T-tubules. Now we're going to look at how do we turn that into calcium release, which calcium release is the trigger for contraction. So let's watch.
Typically, a single motor neuron arising in the brain or spinal cord conducts action potentials that travel to hundreds of skeletal muscle fibers within a muscle. The sequence of events that converts action potentials in a muscle fiber to a contraction is known as excitation-contraction coupling. If we look at a single muscle fiber,
how cool are these models? Look at that. So here, these two yellow walls on the outside, that's sarcolemma. So this is a single muscle fiber. And you can see the pores that enter the T-tubules, which is pretty cool. So the action potential, as it spreads from the neuromuscular junction, is going to go down these pores and down these T-tubules.
We see that an action potential travels across the entire sarcolemma and is rapidly conducted into the interior of the muscle fiber by structures called transverse tubules.
I guess I should have mentioned what the T stands for. Sorry. The transverse tubules.
Transverse, or T-tubules, are regularly spaced enfoldings of the sarcolemma that branch extensively throughout the muscle fiber. At numerous junctions, the T-tubules make contact with a calcium-storing membranous network known as a sarcoplasmic reticulum, or SR.
Okay, so right now at rest, because the muscle's not contracting right now, all of the calcium is stored within the sarcoplasmic reticulum. That is typical of a muscle at rest, okay, because calcium release into the cytosol causes muscle contraction. So that's really the goal of excitation-contraction coupling. It's to take the electrical signal, that is the action potential, and cause calcium release from the sarcoplasmic reticulum. If we release calcium from the sarcoplasmic reticulum, it's going to trigger muscle contraction. Okay, so that is what excitation-contraction is. So let's watch how that happens.
Where it abuts the T-tubule, the SR forms sac-like bulges called terminal cisternae. One portion of a T-tubule plus two adjacent terminal cisternae is known as a triad. The membranes of the T-tubule and terminal cisternae are linked by a series of proteins that control calcium release. As an action potential travels down the T-tubule, it causes a voltage-sensitive protein to change shape. This shape change opens a calcium release channel in the SR, allowing calcium ions to flood the sarcoplasm. This rapid influx of calcium triggers a contraction of the skeletal muscle fiber. Thus, calcium ions are responsible for the coupling of excitation to the contraction of skeletal muscle fibers.
All right, so I mean, we're almost there. We've almost traveled the distance between, hey, I want to lift this heavy thing to, hey, I'm lifting this heavy thing, right? So we've watched the action potential come from the brain and come all the way down to the axon terminal and trigger acetylcholine release into the synaptic cleft, which triggers calcium channels to open in the sarcolemma, which causes the formation of an action potential in the sarcolemma. That action potential then spreads, it travels down the T-tubules, and it opens voltage-gated channels within the terminal cisternae, which, when those open, cause calcium release from the terminal cisternae into the the sarcoplasm, into the cytosol of the muscle cell. We're almost there. Now we just need to see, how does calcium cause myosin and actin to tug on one another? What actually causes what's called cross-bridge formation and then cross-bridge cycling? So that's step 3. It's probably the most confusing of the 3. So bear with me. It's not that bad. And honestly, it's pretty interesting. So let's check.
The contraction of a skeletal muscle generates the force necessary to move the skeleton. A contraction is triggered by a series of molecular events known as the cross-bridge cycle. In a skeletal muscle fiber, the functional unit of contraction is called the sarcomere.
So let me pause it right there. Remember talking about striation?
The sarcomere.
Overlap? So remember, the striation is due to the light bands are where there's only thin filaments. And then the dark area is where thick and thin filaments overlap. So it's pretty cool that when the Z discs come together, the thick filaments actually get bigger. And the thin, well, really what I should say is the thin filaments, the I bands get smaller. So it's
Contraction is called the sarcomere.
So you can see as the Z discs get closer together, the I bands disappear. And that's because there's a greater degree of overlap between the thick and thin filaments. All right, so quick explanation of where we are. These big red things are the thick filaments made of myosin molecules. You can see 2 Z discs on either side, so this defines the boundaries of the sarcomere. The sarcomere is all of this area in here. It's the smallest contractile unit of a muscle fiber. These rods right here are thin filaments, which are made up of actin, tropomyosin, and troponin. And that's going to be the focus. We're going to look at the interaction between the myosin heads and the thin filaments.
A sarcomere shortens when myosin heads and thick myofilaments form cross bridges with actin molecules and thin myofilaments.
Okay, so this nice narrator is probably going to say all this, so I'm sorry for repeating her. But these yellow spheres right here, those are actin subunits. Every single actin subunit has what's called a myosin binding site on it. However, it's covered by this green structure here, which is called tropomyosin. If we expose the myosin binding site, myosin and the myosin binding site, they have high affinity. They're going to bind. All we have to do is uncover that. So to make a muscle contract, really all we have to do is uncover the myosin binding site. So we're going to look at how calcium causes troponin, which is the blue molecule, to change shape and move tropomyosin out of the way. Once that happens, the cross-bridge cycle just automatically starts.
The formation of a cross-bridge is initiated when calcium ions, released from the sarcoplasmic reticulum, bind to troponin. This binding causes troponin to change shape. Tropomyosin moves away from the myosin-binding sites on actin, allowing the myosin head to bind actin and form a cross-bridge. Also note that the myosin head must be activated before a cross-bridge cycle can begin. This occurs when ATP binds to the myosin head and is hydrolyzed to ADP and inorganic phosphate. The energy liberated from the hydrolysis of ATP activates the myosin head, forcing it into the cocked position. A cross-bridge cycle may be divided into...
Okay, so here's where it gets a little bit confusing. We're going to now look at the steps of the cross-bridge cycle. So we saw what initiated contraction. Exposing the myosin binding site caused cross-bridge formation. That's the initiation of contraction, but now we need to see, how does the myosin head actually change shape and tug on the thin filament? And that's what we're going to do here. And so we're starting kind of midway, because we saw that something already had to happen to get to this step. ATP had to bind to the myosin head and be hydrolyzed. And through that process of ATP hydrolysis, energy is released, which causes loading of the myosin head. The analogy to this that I would think of would be loading a catapult. So you have to put energy into the catapult not when you fire it, but when you load it, right? So you have to like, I don't know how it works, but I'm assuming you have to crank it or something, and you crank it, you load it. I guess another example would be like a bow and arrow or a crossbow, even better. So you have to pull back on the string and cock it, right? So now it's ready to fire. So the work was done to load the crossbow, not to actually shoot the crossbow. To shoot the crossbow, you just release all that stored energy. It's the same thing here. So ATP doesn't cause muscle contraction. It causes loading of the myosin head. It actually detaches the myosin head from the myosin filament, and then it hydrolyzes and loads it. So this myosin head right now is in the loaded position. Okay, so we'll see what events have to take place for it to then bind, contract, and then reload. And it's that process that's called a cross-bridge cycle. Bind, contract, reload. Bind, contract, reload. That causes your muscle to shorten. So let's watch it. I'll try not to interrupt.
To 4 steps. Step 1. Cross-bridge formation. The activated myosin head binds to actin, forming a cross-bridge. Inorganic phosphate is released, and the bond between myosin and actin becomes stronger. Step 2. The power stroke. ADP is released, and the activated myosin head pivots, sliding the thin myofilament toward the center of the sarcomere.
Okay, so what causes the myosin head to pivot and cause muscle shortening? It's actually ADP release that does that. Okay, ATP binding causes detachment, hydrolysis causes loading, and then ADP being released causes contraction.
Step 3. Cross-bridge detachment. When another ATP binds to the myosin head, the link between the myosin head and actin weakens, and the myosin head detaches. Step 4. Reactivation of the myosin head. ATP is hydrolyzed to ADP and inorganic phosphate. The energy released during hydrolysis reactivates the myosin head, returning it to the cocked position. As long as the binding sites on actin.
Whoever made this animation, this must have taken forever. Look at this. What's so amazing about this is this happens anytime you wiggle your index finger, right? All of this, any muscle, me talking right now. This is all going on. So it's kind of fascinating to me. But
Actin remain exposed, the cross-bridge cycle will repeat.
Okay, that's the part I wanted to get to. So basically what she just said is that as long as calcium is present, the myosin binding site remains exposed, and this process will repeat. We'll be producing tension. Even if you're just flexing your muscle, this process is going on. It may not be shortening, but the myosin molecules are tugging on the thin filaments.
And as the cycle repeats, the thin myofilaments are pulled toward each other, and the sarcomere shortens. This shortening causes the whole muscle to contract. Cross-bridge cycling ends when calcium ions are actively transported back into the sarcoplasmic reticulum. Troponin returns to its original shape, allowing tropomyosin to glide over and cover the myosin-binding site on actin.
All right, I think that's good. So, I mean, what's just so crazy to me is just do a quick flex of your biceps, like as quick as possible, like a muscle twitch. All of this stuff we just watched happen in that amount of time, from video 1 to the end of video 3. So it's pretty amazing. So guys, I think we'll leave it there for the videos. Go back through and watch them. These are all available. The titles are right here, and then you can always rewatch my video that I'm going to post. And I'm just looking for key events. So picture questions like, what causes detachment of the myosin head from the myosin binding site on the actin subunit? The binding of ATP. What causes the loading of the myosin head? That is the hydrolysis of ATP to ADP and inorganic phosphate. What causes the power stroke? ADP release. What causes detachment? ATP binding, that kind of a thing. And then, of course, what is that initial trigger for cross-bridge cycling? It's calcium binding to troponin, troponin changing shape and moving tropomyosin out of the way so that the cross-bridge can form. Okay, so that's really the depth that I want you to know. So if you need to rewatch a few times and just kind of jot that stuff down, I write my tests from the top of my head. I write new ones every year, and I write them just like that with those kind of questions, which I hope you guys felt from the midterm was fair. So expect more of the same on the final. So let's go back to the PowerPoint. Welcome back, you guys. Oh my goodness. I just listened to that audio. It was so bad. I'm sorry. I would just redo it right now, honestly, but I can't wait any longer to get this posted. We're going to be having lecture and lab practice tests next week, and it's already the weekend, so I just wanted to get this out. I will I will try to get a better version of that posted. But anyway, um, I tried to level it out as much as I could. I couldn't get rid of the echo. I figured out how to get rid of the echo for next time, but for now, it is what it is, and I'm so sorry about that. So um, all we can do now is go forward. So we're on slide 24. We've got to get, oh boy, 54. So I don't want to waste any more time. Let's get going. So real quick, three components of the neuromuscular junction: axon terminal, synaptic cleft, sarcolemma. What's the final trigger for contraction? That is ADP release from the, well, it depends. What do we consider final? What do we consider initial? So ADP is released and the muscle shortens or tension is produced. So to me, that's the final trigger. What is the initial trigger? Calcium binding to troponin. But you could argue that it's action potential formation in your brain or spinal cord too. So I won't ask that question on a test. Um, this is a good question. What prevents the filaments from simply sliding back to their original position every time a myosin cross bridge detaches? Well, there's millions of these cross bridges, so just because one detaches doesn't mean the other, however many, have detached, so it doesn't slide back. What would happen if a muscle fiber suddenly ran out of ATP during partial contraction? I ask this in class, and oftentimes people say, oh, your muscle would go limp, it would relax. Actually, the opposite is true, because remember what ATP does. ATP causes cross-bridge detachment. So it actually would cause, if you run out of ATP, it would cause contraction, or at least maintain contraction, which is what happens when someone or something dies. There's rigor mortis for a while, and eventually that goes away. But anyway, okay, so we got to get cruising, guys. So types of muscle contraction. Okay, first we need to understand the difference between tension and load. Tension is the amount of force produced by the muscle. The load is the amount of weight that force is working against. So let's say you're trying to pick up a 10,000-pound semi truck. No matter how much no matter how much tension you produce, the load isn't going to move. So that would be considered isometric because you're producing contraction force, but there's no movement occurring. Versus isotonic, which is movement is occurring. And whether the muscle shortening or lengthening is dependent on how strong the contraction is. So if we do a strong contraction here, the weight will go up. And we can produce still a contraction, but a weaker contraction, and the weight will slowly lower. So isometric, I don't know if you guys, I'm assuming most of you have been to Disneyland. We always get a picture of my daughter lifting up that weight in Toontown that's bolted to the ground. Do you guys know what I'm talking about? And the bars are made out of rubber, so the bar bends. That's kind of an example of an isometric contraction. Um, Isotonic can be concentric or eccentric. So what that means is if the weight is moving this direction, in other words, the muscle is shortening, that's a concentric isotonic contraction. But if we're producing tension that's slightly less than the load, the weight will actually lower. It's called an eccentric isotonic contraction. We'll come back to that. This next concept is pretty important to understand. So a motor unit is one neuron and all of its innervated fibers. So it's not a one-to-one ratio. Look at this motor neuron right here, this blue one. It connects to, this muscle fiber, maybe this one, maybe this one over here, right? So it's one neuron connecting to multiple motor units, whereas this one is connecting to, this one, this one, this one, this one, this one, right? So motor units are always one motor neuron, but it can be any number of muscle fibers. So if say one motor unit connects to 4 muscle fibers, that'd be a really small motor unit versus maybe a motor neuron going to our glute muscle could connect to hundreds of muscle fibers. So motor unit, the smaller the motor units, the more finely tuned the muscle is going to be. It can produce many different grades of contraction. So the muscle with the smallest motor units, what do you think it is? Picture something. Most people say hands or something, or that's true. Hands are up there. Your finger, your small intrinsic hand muscles. But actually, the muscles with the smallest motor units are the muscles that move your vocal cords, because just a tiny fraction of a movement in one direction can change the tone and pitch of your voice. Versus a muscle with very large motor units might be your glutes. You don't need finely tuned glute muscles, right? It's just maybe 1,000 different contraction forces is probably enough versus 100,000. So motor unit size has to do with how many muscle fibers are connected to each motor neuron, and they will vary. So let's biceps, for example. We have smaller motor units, and we're going to use those when we're lifting, distinguishing between the weight of 1 paper clip and 20 paper clips, right? We need that fine level of coordination, but then we also have motor units that are 1,000 muscle fibers big that help us, lift something between 5 pounds and 10 pounds. So it's all about grading the contraction force. That's where motor units come in. A muscle twitch is not something physiologically, it's something that's done in a lab, and it just kind of illustrates how muscles work. So basically, a twitch myogram is the motor unit's response to a single action potential from the neuron. So phase 1 is the latent period, phase 2 is the contraction period, and phase 3 is the relaxation period. The latent period is all those videos we just watched up into cross bridge formation. It takes time for the action potential to travel, for it to generate in the sarcolemma, for calcium to be released. That's the latent period. Then we have cross-bridge cycling, that's phase 2, that's where contraction is produced, and then we have relaxation. That's the time that the calcium is being pumped back into the sarcoplasmic reticulum. Okay, so how do we grade a muscle response? How do we lift 1 paper clip versus 30 paper clips without throwing them up into the air, right? So we can grade muscle contractions in 2 ways. Number 1, by changing stimulation frequency. That refers to how many action potentials per second we send to the muscle. So if we lift something small, we might send 10 action potentials a second. Something big, we might send 100. So that's frequency of stimulation, and that is called temporal summation. So what that means is summation of stimulus over time. And this graph down here kind of shows that idea. So here's 1 action potential, and we just get a little contraction strength. Here's 3, one after another. So we get a higher peak contraction force. Here's 5, one after another. We get it even bigger. And then here's as many as possible. We get the biggest contraction force. So don't worry about infused tetanus. So number 2, by changing the strength of stimulation. So this is recruitment. How many motor units do we actually recruit? So if you can see down here, these little yellow circles are illustrating, they turn gray when that particular muscle fiber is being recruited. And you can see that the more muscle fibers that are contracting, the stronger the contraction is. So we can recruit more stimuli. We can recruit more motor units. So recruitment and frequency of stimulation is how we grade muscle responses. So that brings us to this idea called the size principle. And this just says that we always activate the smallest motor units first, the smaller and the slowest motor units. And that keeps us from throwing a paperclip up in the air, right? If we just turned our muscle on full strength trying to lift a paperclip, we wouldn't have a whole lot of coordination, right? So we always recruit the smallest motor units, And then we recruit bigger and bigger and bigger motor units and muscle fibers until we achieve the tension that we need, and then we stop. Okay? This also allows us to prevent fatigue. So imagine you're holding a grocery bag at 90 degrees of elbow flexion. Typically, you wouldn't do that, right? You'd let it hang down at 180 degrees. But say you're holding it at 90 for some reason. It may only take 30% of your muscle, of your motor units to hold that back. But you don't just keep the same 30% activated, you shift it around. So say you're recruiting 30% of muscle fibers, they start to get tired, you start recruiting a different 30%, and then let that 30% that was contracting relax. So it allows you to prevent fatigue due to asynchronous contraction. We've already talked about this. So isotonic is a contraction where muscle length changes. If it's concentric, the muscle shortens. If it's eccentric, the muscle lengthens. Compare that to an isometric contraction where there's no change in muscle length. Okay, but there's still a force produced. All right, what is a motor unit? One motor neuron and all of its innervated fibers. So one motor neuron and all of the muscle cells that it's connected to. What's happening in a twitch during the latent period? We covered that. We talked about, action potential has to travel, acetylcholine has to be released, a new action potential has to be formed, it needs to travel down the t-tubules, calcium needs to be released. So all of that, there's a latent period. Matt is competing in a chin-up competition. What types of contractions are occurring in his biceps? So biceps and brachialis cause elbow flexion. So as I'm lifting myself up to the chin-up bar, or as Matt is, that's a concentric isotonic contraction. Okay, and then say I hold myself up over the bar and I look around at my competitors and give them the eye to let them know that I am going to win. That's isometric because I'm just hanging there. And then I lower myself down. That's an eccentric contraction. All right, muscle metabolism. Oh my goodness, look at all these slides we still have. All right, hang in there, guys. So we're going to look at 3 ways that muscles produce ATP. And here they are. We have the creatine phosphate pathway, the anaerobic glycolysis pathway, and the aerobic pathway. All of these pathways produce ATP, and ATP is the only energy source used for contraction. Your muscles store about 5 seconds worth, so the first 5 seconds we can just run on available ATP, but after that we need to replace it. So we need to convert ADP into ATP as quickly as possible. So we're going to look at the 3 pathways that do that. Let's start with the direct phosphorylation pathway or creatine phosphate pathway. So basically this is just a simple reaction where creatine phosphate and ADP react to form ATP. So a phosphate is taken from creatine phosphate and donated to ADP, so now ADP is ATP. This pathway is only viable for roughly about 15 seconds. So we have 5 seconds of ATP storage, then we have another 15 seconds of the creatine phosphate pathway. But as soon as we stop utilizing this pathway, it replenishes somewhat quickly, so you can use it again. So what I mean by that is picture weightlifting. So picture you're doing a bench press. You're pushing that weight up, and then you're taking somewhat of a break. You're still using ATP, of course, but you're taking somewhat of a break at the top, and then you're lowering it down, and then you're pushing it up. Or picture a bicep curl. You're curling the weight, you're resting at the bottom. You're curling the weight, you're resting at the bottom. That is ideal. That's the ideal type of activity for this pathway. You'll probably start utilizing some of the other pathways as well. I mean, of course, you're always doing aerobic respiration and anaerobic glycolysis, but this pathway can be the primary fuel source for that type of activity for some time, maybe the entire session, depending on how hard you're working and how many breaks you're taking. So anyway, what I want you to remember is just what the pathway is, number 1, that it's available immediately. So as soon as that 5 seconds runs out, you can tap into this pathway, but it is only viable for about 15 seconds. Now, there's no oxygen involved in this pathway, So it is technically an anaerobic pathway, but don't let that confuse you with this next pathway that's called the anaerobic pathway. Okay, it's not the only one. This one's just called the anaerobic pathway, and it's often also called glycolysis. So basically, what happens is glucose is broken down into two pyruvic acid molecules. That process releases 2 ATP. Now, this is nice because pyruvic acid is the fuel for the next pathway. So this pathway actually not only produces a little bit of ATP, but it produces the fuel for pathway 3. So glycolysis occurs no matter what, whether oxygen is around or not. So this anaerobic thing can be a little confusing because they think, oh, it's only when no oxygen is available. No, this can happen in both conditions, but it's only viable for about 30 to 40 seconds. Okay, and then if oxygen is present and there's a blood supply, that pyruvic acid byproduct will enter the mitochondria to fuel the aerobic pathway, which is pathway 3. However, at 70% maximal contractile activity, blood vessels are compressed, so the aerobic pathway is not viable. So under these circumstances, most of the pyruvic acid is converted to lactic acid. So let's look at pathway 3. If you're a former bio major or something, you're going to laugh at how quickly we do this. We're just going to hit on some of the high points of the citric acid cycle or the Krebs cycle, and what we call aerobic respiration. So during rest, light, and moderate exercise, this pathway provides 95% of our ATP supply. That's pretty crazy. It occurs in the mitochondria, and its really only weakness, if you could call it a weakness, is that it requires oxygen. So during a maximal contraction, this is not a viable pathway. Basically, glucose or pyruvic acid, depending on where in the pathway we're looking, plus oxygen is converted to carbon dioxide, water, and 32 ATP molecules. So it makes a lot of ATP. It's very efficient. Honestly, that's why if you're trying to lose weight, aerobic exercise may not be the way to go, because when you utilize aerobic respiration, that's a very efficient way to convert fuel energy, glucose, into ATP. And if you're trying to lose weight, you're trying to be inefficient. But anyway, some downsides here. It is the slowest of the 3 systems, so it takes a little while to ramp up. But again, it is very efficient. So the fuel source progression is at first you'll use muscle glycogen, then you'll tap into blood-borne glucose, pyruvic acid, fatty acids. After 30 minutes, fatty acids are the primary source of fuel. This discovery is what kind of led to every treadmill in the world having like a fat burning setting, which was just a slow setting. We now know that that's not really the way it works. Whatever fuel you're using, whether it be carbohydrates, protein, fat, if you're burning more energy than you're consuming than you're eating, you lose weight. It doesn't matter which fuel source you're tapping into. What matters is that you're being inefficient. That's why all these high intensity exercises are popular now. Anyway, so key points for this: oxygen is necessary. That's why it's called the aerobic pathway. It occurs within the mitochondria. It is very efficient, but it's slow. So let's talk about aerobic endurance and anaerobic threshold. So I don't want to mess this up. I have a tendency to mess this up, so hopefully I don't. I'm trying to remember my story that I tell every year. It's a story about a date I went on in college, and hopefully my wife doesn't listen to this. She won't, trust me. So basically, I was trying to date this girl who was on the cross country team. So she was a runner, and I was not a runner, although I was in the best shape of my life. so it's sad that this still went down the way it went down, but a much better shape than I'm in now. But I still was not running marathons routinely like she was. So anyway, after extensive persistence and flirting, I got invited to go on a run with her. So I thought, okay, this is great. I can run and just show how good shape I'm in and talk, and this is going to work out well. So we start running, okay? and we're talking. Things are going well. We're running for about 10 minutes, which I don't know about you. That's about as long as I ever want to run. 10 minutes. So anyway, then she turns to me and goes, "Hey, do you want to start?" And I'm kind of like, "What do you mean?" We started 10 minutes ago. And she goes, "No, do you want to start running?" I'm like, we're literally running right now." I didn't say that. I just said, "Sure, yeah, let's start." So then we like triple our speed. I thought I was running. Apparently, I was jogging at a brisk pace. So then we start running. So up until that point, I certainly was within my aerobic capacity. I was using my aerobic pathway because one key indicator of whether or not you're using aerobic pathway is how hard you're breathing. So I was still able to converse. So what that meant is that my body was not dying for oxygen. It was getting enough oxygen to fuel my aerobic pathway. Then we started running faster, and now it's getting hard for me to talk. Not for her; she's still talking to me, and I'm just like, I'm nodding, smiling. Um, I'm kind of gasping for air at this point, trying not to look like I'm gasping for air, but I'm gasping for air. And that's because now starting to bump into the limit of my aerobic endurance. My body is trying to increase the amount of oxygen that I'm bringing into my blood. So I'm breathing harder and harder and harder. And then at some point, as you increase either duration or intensity of the exercise, something changes. So, I mean, go out and just slowly start increasing the speed of your running. You can almost feel once you've exceeded your aerobic endurance and you've crossed into your anaerobic threshold. What that means is my aerobic pathway can no longer support the demands that I'm placing on my ATP supply. So I've now crossed into my anaerobic threshold. Okay, so I'm now utilizing glycolysis as well as aerobic endurance. Potentially, depending on how fast I'm running, if I'm really sprinting, I might have switched completely over to glycolysis because not enough blood can get to my muscles. And what do you guys know about the anaerobic pathway? It's only good for about 30 to 40 seconds. So now I'm in a bad place. I've got 30 to 40 seconds left at this pace. So then you start kind of running funny, right? You're like skipping, you're running sideways, you're trying to use any different muscle you can. And no matter how much willpower you have, you just have to slow down. So that's when you go, hey, let's slow down here for a second. And there's shame, of course. And anyway, then you have to slow down. So, uh, and then you have to, what? You have to drop back down to a level of intensity that's within your aerobic endurance, within your aerobic pathway. So, anyway, it was okay. She told me later that she was kind of messing with me because we had ran a lot. But anyway, wasn't meant to be. So I hope that goes some small way to explaining these two things. Um, Basically, sitting here talking to you right now, I'm utilizing my aerobic pathway. At some point, though, the intensity becomes so high that the aerobic pathway isn't enough, and you cross into an anaerobic threshold. Okay. And then you can only maintain that high intensity for 30 to 40 seconds before you have to decrease your intensity or you just fall down. there's no way around it. You can't fight science in that regard. So some examples of when you might use each of these pathways: weightlifting, you're going to use your creatine phosphate or direct phosphorylation pathway. High intensity on off activities such as tennis or soccer or 100-meter sprint swim, you're going to be utilizing your anaerobic pathway. Prolonged jogging, walking, sitting, you're going to utilize your aerobic pathway. Oh, I kind of have to sneeze. Okay. So what is muscle fatigue? Muscle fatigue, physiological muscle fatigue, is not just like, oh, I'm so tired, my muscles are sore. It's a physiological inability to contract in the presence of stimulus. So what that means is action potentials are being sent to your muscle, but your muscle will not contract. And that's caused by such a high level of exercise intensity and duration that the ion balances that exist at the membrane potential that allow excitation contraction coupling are absent. So much sodium's been allowed in, so much potassium's been allowed out. so much calcium's been absorbed by the axon terminal that you physically cannot cause a muscle contraction. So that's pretty rare. If you've ever watched Iron Man or some of those crazy triathlons and people are like falling down at the finish line, that's muscle fatigue, true physiological muscle fatigue. EPOC, or oxygen debt. This just refers to the fact that after you exercise, muscle tissue must replenish its oxygen reserves bound to myoglobin. So even after you're done exercising, your oxygen intake and conversion to CO2 is going to be higher at rest than it was at rest before exercise. So you don't burn all your calories during the exercise itself. There's an oxygen debt, or a post-exercise oxygen consumption. So this increased oxygen demand during the recovery period is referred to as EPOC, or oxygen debt. What else do muscles do? They produce heat. Only about 40% of the energy released from ATP during muscle contraction is converted into tension. 60% is converted into heat. Now you could say this is an inefficiency, that's a design flaw, or you could say this is a amazing way to maintain our body temperature, and I'm sure it's the latter. So this inefficiency produces heat, which maintains our core body temp. Clayton has just finished jogging and is breathing heavily. Why is he breathing heavily? He's breathing heavily to bring in more oxygen so that he can utilize his aerobic pathway as best as possible, because the anaerobic pathway is short-lived. So we're always trying to use the aerobic pathway to the best extent that we can. What metabolic product might account for his sore muscles? So this comes out of your book. This is kind of an ongoing debate, I think. I haven't looked into it for a few years, but last I looked, people were starting to think that lactic acid is not the cause of muscle soreness. The edition of your book I have is several years old, though, so it refers to lactic acid being the cause of muscle soreness. Nowadays, more and more people are thinking that it's actually microtrauma at the muscle fiber level that causes soreness. That may be a debate that's been figured out already, but as of a year or two ago, it wasn't. So now we're going to look at forces of muscle contraction. Forces of muscle contraction. We're going to describe factors that influence the force and velocity and duration of contraction. So we're in the home stretch here. So if you're just hanging on by a thread, don't worry. We're just about done, and I'll have this posted here in about an hour, hopefully. So muscle contraction force, influencing factors, recruitment. So we already know that. The number of fibers recruited is obviously going to affect the tension production. The size of the muscle fibers is going to affect the tension production. The frequency of stimulation and the degree of muscle stretch. I want to touch on this because it comes back when we get to the heart chapter. So the degree of muscle stretch. If you've ever gone to a gym and stood in front of one of those big floor-to-ceiling mirrors with your bicep curl weights by your side, you've experienced this. Especially if it's, I hope this isn't sexist in any way, but especially if you're a guy, you've done this. Maybe everybody does it, I don't know. I can only speak for myself. How about that? So you grab the weights, and maybe like the 30-pound weight would be the right weight for you to grab, for your physique, but you grab the 40-pound weight because you're in public, and there's a pride factor. So you grab the 40-pound weights and you go stand in front of the mirror, and you have overdone it. The 40-pound weight is not the weight for you. So what do? I wish you could see me. So you have the weight hanging by your side, right? We're doing a bicep curl. You do a little bit of a swing. Like you kind of swing the weight back and swing it forward, because that first like 10, 20, 30 degrees, you're weak right there. And then you get kind of in the mid-range, and you could lift anything. You could lift 50 pounds right there. But then you get closer to the top and you start struggling again, so you kind of lean back and let the weight fall backwards to the top against your shoulder, right? I hope you can visualize that. But anyway, anyone who's done a bicep curl with more weight than they should knows what I'm talking about. There's probably other examples, like a shoulder press. You might bend your knees and kind of stand up to get it up off your shoulders. The point is, there are optimal muscle lengths for force production. And for skeletal muscles, it's mid-range. Mid-range is the optimal spot, because picture this. So here's your thick filament, how do you draw a thick film here? Here's your thick, and then here's your thin filament. Okay, and you have these little myosin heads attached. Okay, so why did I draw it like that? Let's do it like so the weight's down by your side. So here's the weight down by your side. There's very little, these are two thin filaments. Here's your thick filament, okay? There's very little overlap. There's just a couple myosin heads, right? So you can't produce much force there. This is the worst drawing I've ever done. But then let's say now the weight's up by your shoulder. It's very high up. Now the thin filaments are kind of almost like bunching up on one another. There's a lot of overlap, but you're almost at the end of your range. So the muscle gets weaker when the thin filaments are really close together, right? So the middle ground where the thin filament maybe stops like right here, that's where your muscles are strongest. So muscles are strongest in mid-range. That's their optimal degree of muscle stretch. What influences velocity? Muscle fiber type, which we'll talk about on the next slide, load and recruitment. So the smaller the load and the bigger the recruitment, the faster the contraction will be. And that brings us to fiber type, because fiber type is based on two criteria, speed of contraction and ATP pathway. So we have three fiber types. We have slow oxidative, okay, fast oxidative, and fast glycolytic. So the first part here, the slow, fast, fast, refers to twitch speed, how quickly the muscle shortens, the fiber shortens. And the second part refers to the major pathway utilized. So slow oxidative relies heavily on the aerobic pathway and is slow. Fast oxidative is a bit faster and still relies on aerobic pathway. Fast glycolytic is the fastest, but utilizes glycolysis primarily. So there's not a lot of mitochondria present, not a high capillary density because they're utilizing less oxygen. We all have sort of a predetermined ratio of these that changes based on what type of sport and activity we do. So if we become a marathoner, we're going to have more slow oxidative. Those are smaller, so your muscle mass actually decreases, but your endurance skyrockets if you're like a cross-country runner, for example. Fast oxidative is kind of in the middle ground, and fast glycolytic, these are the fattest muscle fibers. So if you're maybe like a weightlifter, for example, you would expect to have a lot of fast glycolytics. Increased load results in longer latency, a slower contraction, and a shorter potential duration because you fatigue more quickly. Recruitment. The more fibers are recruited, the faster the contraction and the more prolonged the contraction. We've got a helicopter flying over, sorry. Adaptations to exercise. Okay, so if you do a lot of aerobic exercise, capillary density increases. The demand for oxygen increases, so your body responds by increasing the blood supply to that muscle fiber. So the number of capillaries increases, the number of mitochondria increases, and the concentration of myoglobin increases. This affects all fiber types. So if you start to do more aerobic exercise, this happens to all of your fibers, and it can happen so much, in fact, that conversion is possible. So fast oxidative may convert to slow oxidative, or fast glycolytic may convert to slow oxidative. So if you start doing a lot of aerobic exercise, your muscle fibers become much more efficient users of oxygen. Your aerobic capacity increases, and your muscle mass will actually decrease. Resistance exercise, on the other hand, causes muscle hypertrophy. Muscle fibers get bigger as they convert to fast glycolytic, especially they get much bigger. They do increase in diameter, but not number. So you don't actually make new muscle fibers. The ones you have just get bigger. And something kind of interesting is that hypertrophy cannot occur in the first 30 days. And when I learned that, I said, no way. Because when I'm like 2 weeks into one of my many started and failed attempts at getting really fit, 2 weeks in, you're feeling buff. You're like looking in the mirror like, whoa, I got to keep this up, right? But that's, I guess it's all in your head because hypertrophy cannot occur in the first 30 days. But you may say, that can't be true because I know for a fact that I was lifting more weight. And that actually is true. So you can start experiencing gains in strength much earlier than 30 days. But that gain in strength is actually due to improved nervous system characteristics. So your nervous system gets better at activating your muscles. So it's not just your muscle tissue that determines how strong you are, it's also your nervous tissue. Nervous tissue responds more quickly, so you are going to experience strength gains fairly early in a resistive exercise program, but you won't experience hypertrophy to roughly the 30-day mark. So just kind of a random factoid. Guys, we did it. That's it. So I'm going to get this posted, and I hope it's not too brutal on you all. The audio problems earlier, I hope you can figure out a way to survive that. I'll do my best to get something posted for sure before the test, but it won't be this weekend. Maybe some individual videos on those YouTube videos. So I'll post a few files to anponline.com. I'm going to go ahead and get this posted on Canvas right now. I'll have an announcement up in the next few days about our practice tests next week. Okay. Oh, and just so you're wondering while you're doing this, the practice test for lecture, which is what this is, a lecture video, is going to be take home. I'm going to post it. You have all week to do it. So don't stress so much about lecture. We will have a lab quiz on Wednesday at 6. So if you're trying to decide what to study for, I'd probably focus on lab first. Okay? Thanks, guys.